Hello, all of you. I hope all of you are staying safe in this period of lockdown. So, today I would like to talk about food microbiology. So, first of all, I would like to give introduction of the food microbiology. So, food microbiology is the study of microorganisms that inhabit, create, or contaminate food, including the study of microorganisms causing food spoilage. Good bacteria, however, such as probiotics, are becoming increasingly important in food science. Microorganisms are involved in food spoilage, food poisoning, and also in food preservation and production. So, most common microorganisms which we will study in food microbiology are bacteria, molds, yeast, and viruses. So, in another words, or in another way, you can define food microbiology as a branch of science or research which generally involve the study of microbes that have both beneficial and deleterious effect on the safety and quality of foods and are thus concern of public health. So most common microorganisms include bacteria, molds, yeast, and viruses. So now I would like to talk about the importance of food microbiology. So the first importance of food microbiology is that it is helpful in finding or studying the food-borne diseases which have microbial origin. So here food-borne diseases caused by consuming contaminated food or drinks. Microbes and toxic substances can contaminate foods. There are more than 250 known foodborne diseases. Majority are infectious and are caused by bacteria, viruses, and parasites. Other foodborne diseases are essentially poisoning caused by toxins secreted by microbes. Second important uh, second important thing which we learn by through food microbiology is that it helps in finding the microbes which generally causes food spoilage. So food spoilage is the process which involves any change which reduces which renders food unacceptable for human consumption. And food spoilage are generally caused by a growth and activity of microorganisms like bacteria, yeast, and molds. Now, the third importance of the food microbiology is that it also helps in studying the uh, beneficial uses of microbes in food. Various microorganisms are essential for the production of food, such as beer, bread, yogurt, wine, and other fermented foods. Fourth importance of food microbiology is that it helps in finding the way to control the microbial growth in food. This is done by regulating the temperature, pH, humidity of the environment. Fifth fifth main importance of the study of food microbiology is that it helps in finding the way to reduce or destruct the microbes in foods. And sixth importance of food microbiology is that it is helpful in understanding the microbial food fermentation. So here question arises that what is microbial food fermentation? So the microbial food fermentation is the process of converting carbohydrates to alcohol or organic acids using microorganisms such as yeast and bacteria under anaerobic condition. Food microbiology is also helpful in studying about probiotic bacteria. So what is probiotic bacteria? Probiotic bacteria are often known as good or helpful bacteria because they keep help in keeping our stomach or gut healthy. You can find probiotics in supplements and some foods like yogurt. So this is all about the importance of food microbiome. Now I would like to talk about foodborne diseases of microbial origin. So foodborne diseases are generally caused by consuming contaminated food and beverages. And many different disease causing microbes or pathogen can contaminate foods. So there are many different types of foodborne illnesses. These foodborne illnesses through microbes are generally caused by the infection spread by the different harmful microbes, and they can also be caused by the toxin released by different microbes which generally grows in different kind of foods. So now I would like to talk about symptoms of foodborne illnesses. So first symptom is diarrhea and vomiting. Second, other symptoms are abdominal cramps, nausea, fever, joint and back aches, and fatigue. So this is the table where in first column different kind of foodborne diseases are given. In the second column, different kind of organisms or microbes which are related to different kind of foodborne diseases are given. Sorry. In the fourth column, you can find all the food where these microorganisms grow, which ultimately leads to the development of these diseases in basically in human body. So by going through this table, you can get idea about different kind of organisms which generally grows in different kind of foods, and when we take these foods, then what kind of disease from which we can suffer. So just go through this table and try to learn three to four important foodborne diseases. So the first important foodborne disease which you will have to keep in mind is salmonellosis. So salmonellosis is generally caused by Salmonella enterica, and Salmonella enterica generally grow in meats, poultry, fish, eggs, and dairy products, right? Second major foodborne disease which you will have to keep in mind for the examination point of view is Archobacter diarrhea and Campylobacteriosis, which are generally caused by Archobacter buchleri and Campylobacter jejuni respectively, and these bacteria generally grows in meat products, especially poultry, milk, pork, poultry product, water respectively. Another important major foodborne disease you will have to keep in mind for your examination point of view is Escherichia E. coli, which causes diarrhea and colitis, and the organism which is responsible for causing this disease is E. coli, including serotype O157H7, and these bacteria generally grow in undercooked ground beef and raw milk. So these are the three major foodborne infectious diseases which you will have to keep in mind for your examination point of view or any competitive examination point of view. Others are important, but you can leave them. Now these are the diseases which are caused by which are generally caused by consuming the consuming the food where these bacteria grow, and these diseases are generally caused by their infection of these bacteria. But now I would like to talk about the diseases which are caused by the bacteria which grows on food and it releases some toxins, right? So bacteria or microbes, bacteria generally generate the the toxin which are generated by bacteria are generally classified into two categories. First is exotoxin and second is endotoxin, right? And exotoxins are generally produced by gram positive bacteria, and exotoxins are generally protein produced inside pathogenic bacteria, and the exotoxin are then secreted or released into the surrounding medium following lysis, right? And the example of the bacteria which generally secrete exotoxins are Bacillus cereus, Bacillus anthracis, Staphylococcus aureus, Streptococcus pyogenes. And the example, another type of toxin which are released by bacteria are endotoxin. And endotoxins are generally released by gram-negative bacteria. And endotoxins are a lipid portion of lipopolysaccharides that are the part of outer membrane of the cell wall of the gram-negative bacteria. And the endotoxins are liberated when the bacteria die and cell wall breaks apart. And the examples of bacteria which releases endotoxins are Salmonella typhi, E. coli, Vibrio cholerae, and Shigella. So the main disease which are generally caused by the toxin, which are generally found in the human body and which are generally caused by the toxin released by the bacteria growth in bacteria which are bacteria growing in foods are staph food poisoning, and it is a type of gastrointestinal illness. And as I already told you earlier, it is caused by eating food contaminated with toxin produced by the bacterium Staphylococcus aureus. So some viruses are also responsible for the development of foodborne diseases in humans. These are rotavirus or Norwalk virus, and second is viral hepatitis A. And generally virus, unlike bacteria, cannot multiply in food, and the main mode of the transmission of these the main mode of transmission therefore is by food handlers or by the use of dirty utensils which transfer the virus to food, whereupon it is ingested by humans. So these kind of bacteria spread through different kind of dirty utensils, or you can say this spread contagiously or through contact. Now the toxins, different kind of toxins which are generally produced by fungi are are known as mycotoxins, and these mycotoxins are also responsible for the development of different kind of diseases in human body. So the most important mycotoxin is aflatoxin, and aflatoxins are generally secreted by Aspergillus flavus, Aspergillus parasiticus, and Aspergillus nomeus and penicillium. And these fungi generally grows in these foods, that is cereals, feeds, oil seeds, and oilseed pulp and coconut. and this toxin help aflatoxin is the important carcinogenic compound, and carcinogenic compound are generally responsible for the development of cancerous cell in the human body. Aflatoxin also causes also responsible for affecting the structure of DNA, or you can say mutating the DNA. It also causes hepatitis, bleeding, and kidney lesions. Another important mycotoxin is fumonism. Fumonism is generally produced by fusarium, produced by the fungi of genus Fusarium, and these fungi generally grows on cereals and corns, and this toxin generally causes encephalomalacia, pulmonary edema, and this toxin is also responsible for the development of cancerous cell in the human body. Similarly, these are the different toxins which are generally released by these different kind of fungi, and these which generally grows in different kind of food product, and this generally causes different kind of diseases in human body. So here I would like to talk about the two most important mycotoxins which you will have to keep in mind for the examination point of view. First is ergot body, which are generally produced by parasitic fungus of genus Claviceps, and it generally cause ergotism, right? Second one is aflatoxin. About aflatoxin, I have already discussed that aflatoxin is the important mycotoxin which are generally produced by Aspergillus flavus, and Aspergillus flavus generally grows in cereals, feeds, oilseeds and pulp and coconut. So this is all about the different kind of diseases which are generally caused by different kind of microbes like bacteria, viruses and fungi. Now I would like to talk about microbial food spoilage. So about food spoilage, I already told you, I already discussed that food spoilage is the process through which any kind of food becomes unfit to consume. So a spoilage of food generally takes place by two kind of factors of a particular food. First is intrinsic factor of the food, and second is the extrinsic factor of the food. Intrinsic factor of the food generally present inside the food, and it generally intrinsic factors are the composition of the food, physical and biological state of the food, and the extrinsic factor of the food is generally present outside of the food, and these are temperature, relative humidity, gases and contaminating microorganisms. So change in both intrinsic factor and extrinsic factor generally encourage or produce an environment which generally encourages the production of harmful microbes in, or the growth of harmful microbes in a particular food, which ultimately changes the intrinsic factor and again the extrinsic factor of the food, and which makes the food unfit to consume, and that process is known as food spoilage or fermentation. So this is all about the microbial food spoilage. Now I would like to talk about something about the different factors which affect the food spoilage. So the first important factor is pH. So as you know that pH is very important for executing a different kind of chemical reaction present in any biological system. So bacteria generally grows at physiological pH, and that physiological pH ranges from pH 6 to pH 7. So when in a particular food, due to some uh, environmental change, pH reaches around pH 6 to 7, then it it provide a very ideal condition for the uh, growth of different harmful bacteria, and it encourages its growth in a particular food, which ultimately leads to food spoilage. Second factor is water. So water is the medium where microbial growth generally takes place. So a food which is rich in more water, or which has more water content, is more prone to bacterial growth and food spoilage in comparison to the food which has lesser water content. Third factor is temperature. So you already know that temperature is also important for the growth of different uh, different biological organism. So at generally physiological temperature, that is between 30 degree to 40 degree, most of the microbes generally grows, right? So in a in a food, if a particular food is if we keep our food in the physiological temperature, that is between 30 degree to 40 degrees C, then you can notice the growth of you can notice the growth of various microorganisms in your food, and which ultimately leads to the spoilage of that particular food. Fourth important factor is the gases. So if a particular food is rich in oxygen, then there is a chance of the growth of aerobic microorganism, and if a particular food is devoid of oxygen, there is a chance of the growth of anaerobic microorganisms, which ultimately leads to its spoilage or fermentation respectively. Now fifth factor is physical composition. So physical composition generally denotes the hardness and softness of a particular food. So hardness generally depends on the uh, lack of water. So if you notice a dry fruit has very less amount of water. That's why there is very less chance of the growth of any microbes in the dried food. That's why the spoilage of dry food is the chances of the spoilage of dry food is very less in comparison to the food which are soft. And here soft food means the food which are rich in the foods which has more water content, right? Now food spoilage is the process where a food product becomes unsuitable to ingest by the consumer. So in this table you can notice that uh, you can notice a food with different kinds of foods, and in this food, what are the substrate which are vulnerable for the spoilage, and as a result of this spoilage, what kind of other products form in this particular food, right? So for example, in fruits, as a result of food spoilage, pectinolysis takes place, and pectinolysis of which substrate takes place, that is pectin, and as a result of pectinolysis or spoilage, methanol and uronic acid, as a result production of methanol and uronic acid generally takes place, and it results in the loss of fruit structure, and as a result of that, soft rot takes place. Similarly, in the case of meat, degradation of protein generally takes place, and as a result of that, formation of different kind of amino acids, peptide and amines generally takes place, which results in the bitterness, souring, bad odor and slimminess in a particular meat takes place. And this takes place these typical products forms as a result of uh, food spoilage of a particular food, that is meat. So these are the different stages of food spoilage. First step is the microbial attack. And what are those microbes? Those microbes are bacteria, yeast and molds. Second step is food degradation. Third step is the changes appear as a result of food spoilage. And what are those changes? Those changes are changes in color, odor, changes in texture, accumulation of gas or foams, or accumulation of liquids. And the fourth step is the enzymatic reaction or decomposition, that is also known as fermentation. Now in this table you can observe the different kind of bacteria and fungi which generally leads to the food spoilage. And in this column you can see the foods, different kinds of foods which are susceptible to these kind of bacteria and fungi, and what are the changes generally takes place as a result of the spoilage of these particular food in response to the attack of these bacteria and fungi. So just go through this table and try to keep all the try to learn or try to keep all these things in your mind, because this table is very important for the for your competitive examination point of view. Now in this slide I would like to talk about how you can preserve your food from microbial food spoilage. So the first step is, so the first thing through which you can preserve your food through microbial food spoilage is by freezing the food. So freezing generally discourages the growth of different microbes, and by this way, by preventing the growth of different microbes through freezing, we can uh, prevent our food from food spoilage. Second way through which you can preserve your food from microbial food spoilage is by boiling. Through boiling you can kill various microbes which are generally used to grow on different kinds of foods. Third way is through adding different kinds of preservatives like salts and salts and sugar sweetening products. And as you already know, as you know that if you will put salts in if you apply salts in any microbial uh, colony or the media which is present, or the media where a lot of microbial colonies are present, then by addition of salt, sorry, if you will add salt in different kinds of microbes or microbial cell, it sucks out all the water or most of the water from the microbial cell, and by this way all the microbes all the microbial cell will be killed due to its dehydration, due to their dehydration. So the fifth step is dehydration. So dehydration is also very important, or you can say it is also very useful way through which dehydration of food, actually dehydration of food is also very useful way to prevent our food product from microbial food spoilage. So the most important example is development of uh, or production of different kind of dry fruit. So in dry fruit, what we do, we generally put uh, different kind of fruits or spices in sun, and as a result of that, most of the water released. And as you know that if in any food there is less amount of water, then that food are less susceptible to the growth of different kinds of microbes. So by this way, through dehydration of a particular food, uh, you can prevent that food from microbial food spoilage. Sixth, uh, Sixth important way through which you can preserve your food from spoilage is by canning. So canning is the process through which you put your food in a jar and keep that keep that jar in high temperature. And after applying high temperature, most of the bacteria will be killed. And in the next step, you cover that jar by removing all the air, or you can say you generally pack that jar by removing all the air and moisture. And if air and moisture is not present in a particular food, then that food can be kept for years without getting spoiled. So these are the 6 ways through which you can prevent your food from microbial food spoilage, or these are the ways of food preservation. So this in this table also you can see the different kind of approaches through which you can preserve your food, and these approaches are removal of microorganisms, applying low temperature, high temperature, reduce water availability, that is dehydration. There are some chemical based preservation method through which you can prevent your food from food spoilage. You can apply radiation to your food. Radiation also helps in killing different kind of harmful bacteria, or it can also prevent the growth of different kind of bacteria which cause food spoilage. Uh, Another way is the microbial product based inhibition, and in this process we generally add substance such as bactericidins to food to control foodborne pathogens. Uh, so this is all about the food preservation method. We generally preserve or prevent food from microbial food spoilage. So these are different kind of food preservatives which generally helps in preserving the food or preventing the food from microbial food spoilage. So the most important food preservative is propionic acid, and it generally helps in preventing these foods, that is bread, cakes, some cheese, from different kind of molds. Second important preservative is benzoic acid or benzoates, and these preservative generally helps in preventing margarine, pickled relishes, apple cider, soft drinks, tomato ketchups, and salad dressing from yeast and molds. And third important preservative is parabens, and when parabens is used to apply uh, in these foods that are bakery products, soft drinks, pickles, salad dressings, it generally prevent these food from yeast and mold growth. So by this way, by adding preservatives, you can prevent your foods from microbial food spoilage. So now, after learning about the harmful effect of bacteria in food production, now I would like to talk about beneficial uses of different microbes in food. So microorganisms are essential for the production of food such as cheese, yogurt, bread, beer, wine, and other fermented food. And fermentation is one of the methods to preserve food and alter its quality. Yeast, especially Saccharomyces cerevisiae, is used to leaven bread, brew beer, and make wine. So there are several foods which are generally produced by, or which are generally developed by use of different kind of microbes in household, right? And what are those foods? So those foods are curd from milk, which are generally produced by fermentation through the bacteria that is known as lactobacillus, and yogurt is generally produced by bacteria that is lactobacillus bulgaricus. Saccharomyces cerevisiae is a type of yeast used for making bread in the household as well as food processing industry. So about food processing industry we will talk in the latter part of our presentation. Third one is yeast is also used to prepare some traditional drinks like toddy by the fermentation of flower sap from coconut palm. In addition to these, most common food like dosa and idli are prepared for fermented rice by some bacteria. And what are those bacteria? Those bacteria are Leuconostoc, mesenteroids, Streptococcus faecalis, Lactobacillus fermentum, and Bacillus amyloliquefaciens. And these bacteria are the prominent bacteria responsible for souring and leavening of dosa batter. So this is all about the bacteria which are generally responsible for household food processing. Now I would like to talk about the bacteria which are generally useful for industrial food processing, or you can say industrial production. So in this series, so in this direction, antibiotics are the important component of human welfare against infection and diseases, and these are manufactured in industry using bacteria and fungi. So here antibiotics are the chemical compound which are generally derived from different kind of bacteria and fungi, and which generally helps in killing or preventing or reducing the growth of different harmful bacteria. And penicillin is one of the important antibiotics, and it is generally produced by fungi that is known as Penicillium notatum, right? And it is a beta lactam antibiotics. Why it is known as beta lactam antibiotics? Because this antibiotic generally has beta lactam. These antibiotics has an important component which is in the cyclic form, which is present in the cyclic form, and this component is known as beta lactam. And beta lactam plays major role in killing the bacteria by preventing the biosynthesis of bacterial cell wall. And penicillin was first discovered by Alexander Fleming in the year of 1928, and for this he got Nobel Prize in Physiology or Medicine in 1945 with Ernst Boris Chain, and Sir Howard Walter Florey. So, so the discovery of penicillin was generally done. Discovery of penicillin was generally done by Alexander Fleming, right, in the year of 1928, and this discovery is known as serendipity. So, as you know, that serendipity is the accidental discovery of particular scientific, excellent discovery of a particular scientific research, right? And there is one small story behind the discovery of this penicillin, and I would like to discuss that small story. So Alexander Fleming was a microbiologist, and on one day he developed a plate with microbial growth medium, right? And in this plate he wanted to grow gram positive bacteria. So he streaked that bacteria in this nutrient medium and kept that plate in overnight in uncovered condition, right? And in the next day when he noticed the plate, he was surprised to see one thing, that in one side he found the lot of gram positive bacterial colonies, but in another side he didn't get any gram positive bacterial colony, but in another side he found the growth of one contamination, and that contamination was of mold of genus Penicillium, and around that mold he didn't get any colony of, or he didn't find any colony of gram positive bacteria. So by this observation he conduct various research work in this direction, and after observing the result of all these research work he came to the conclusion that this mold generally produces a compound which generally kills the gram positive bacteria, right? And that's why at the initial stage of its experiment he didn't get any colony of gram positive bacteria around this mold. So by this way an antibiotic known as penicillin was discovered. And as I already told you earlier, that the penicillin is generally effective against gram positive bacteria, and it is commonly acts as bactericidal agent, causing bacterial cell death by inhibiting bacterial cell wall biosynthesis. So this is all about the discovery of penicillin by Alexander Fleming. And this is the table, and in this table you can find the antibiotic, and from where these and this is the, in this column you can find the various organisms from where these antibiotic have been derived, and in the third column you can find the different kinds of bacteria on which these antibiotics acts, and in the fourth column you can find the site where these in the bacterial cell where these antibiotics act. So this is very important table, and try to learn this table very seriously, because lots of question are usually asked from this table in various competitive exams. So the important one is penicillin, erythromycin, neomycin, streptomycin, gentamicin and rifamycin. Now, apart from the antibiotics which are which are derived from microbial, which are derived from different kinds of microbes, there are some natural antibiotics also, and these natural antibiotics are uh, allicin, which are generally derived from garlic, methyl glycol, that is, which are generally derived from manuka honey. Similarly, from there are total 11 plant products from where these secondary metabolites are generally produced, which generally acts as antimicrobial agents. So these antimicrobial agents generally acts as natural antibiotics, right? Just keep in mind, just remember the names of these plants, and you will have to remember the product or component which are generally derived from these plants, which has antimicrobial activity. Now, the second thing, which the second important food product which we generally uh, derived from the use of different kind of microbes are beverages like whiskey, brandy, beer, and rum, and these are derived from the process that is known as fermentation, and the microbes which generally takes part in this fermentation is Saccharomyces cerevisiae, right? And these are the alcoholic drinks which are generally derived from the fermentation of these materials, and the fermentation process was generally performed by the microbe that is Saccharomyces cerevisiae. And these alcoholic drinks are beer, wine, whiskey, brandy, rum, gin, vodka, and tequila, and these are derived from the fermentation of barley malt, fruit juices such as grapes, fermented cereals, fruit juices, sugarcane byproducts such as molasses, rye malt, potato agave, respectively. And fermentation is the chemical process by which molecules such as glucose are broken down anaerobically. So this information is also very important for all of you, especially those who are preparing for the JRF in plant science stream. So there are some another important fermentation product like ethanol, which are generally derived by the which are generally used as industrial solvents and beverage, and the organisms which are generally involved in the production of ethanol is Saccharomyces cerevisiae. Glycerol again is generally produced by the use of Saccharomyces cerevisiae. Lactic acid again are generally produced by the fermentation process through Lactobacillus bulgaricus. Uh, Clostridium acetobutylicum is generally used for production of acetone and butanol, and Bacillus subtilis is an important bacteria which generally helps in the production of alpha amylase, and it is generally used in the hydrolysis of starch. So just remember this table again. Just try to remember this table. Any question can be asked from here in your competitive exam. So microorganisms are also involved in commercial production of enzymes. Example, production of lipase and alpha amylase. About alpha amylase, I have already explained. Again, about ethanol, I have already explained. So microorganisms are also responsible for the formation of immunosuppressive agent like cyclosporin, and it is generally produced by one fungus that is known as trichoderma. So suppressive agent may be given to prevent the body from forming an immune response to an organ transplant or to treat a disease that is caused by overactive immune system such as rheumatoid arthritis. So in various autoimmune disease, these cyclosporins are uh, very useful. So in food processing technology, some of the microorganisms are used for the preservation of packed food as well. So this is all about the use of different kind of microbes in food and industry. So, and this is all about the chapter that is food microbiology. So if you have any problem in understanding anything regarding this class, you are free to or you please feel free to contact with me. So thanks to all of you. Bye.